What if your family tree wasn't just a few generations, but stretched back tens of thousands of years to forgotten continents? Prepare to unlock the incredible secrets hidden within your own DNA, revealing the epic journey of humanity and your personal connection to ancient civilizations you never knew existed. Welcome to our channel, the place where knowledge meets curiosity. Here, we dive deep into the wonders of history, science, culture, and mysteries that shape our world. Every video is designed to open your mind and spark your imagination. So sit back, relax, and let's begin this incredible journey together. Now, we explore, explain the human genome, unlocking our ancient ancestry. From the moment the first humans gazed up at the night sky, questions about where we come from have echoed through time. We have traced our ancestry through oral traditions, carvings, and bones buried deep in the earth, but never before have we held the true key to our identity within our very cells. The human genome, the complete set of genetic instructions encoded within DNA, is more than a biological map. It is a chronicle of evolution, migration, and survival. Every gene, every nucleotide is a record of ancient struggles, triumphs, and adaptations that tell the story of how we became who we are. To unlock the human genome is to open a time capsule that reaches back millions of years, connecting us not only to our human ancestors, but to every living thing that has ever existed. In 2003, the Human Genome Project announced the completion of a monumental task, decoding the roughly 3 billion base pairs that make up human DNA. This was not merely a scientific milestone, but a turning point in human self-awareness. For the first time, we could read the script of life written in 4 simple letters: A, T, C, and G. Yet, as scientists soon realized, understanding the code was only the beginning. The real question was what this code could tell us about who we are, where we came from, and why our species evolved in the way it did. The genome became not just a biological puzzle, but an anthropological and historical one. The DNA molecule, elegantly twisted into a double helix, is the most efficient storage system ever conceived by nature. Within each of our trillions of cells lies an entire library of information, passed down generation after generation, edited and rewritten by time itself. Every mutation, every duplication, every silence in the code is a clue, a whisper from the past. It tells of migrations across continents, encounters with other hominins, battles against disease, and the slow shaping of intelligence, creativity, and emotion. The genome is not static. It is a living manuscript that continues to evolve as humanity itself changes. The story of our genome begins long before Homo sapiens walked the earth. More than 3.5 billion years ago, the first self-replicating molecules emerged in the primordial oceans, perhaps sparked by lightning or mineral reactions at hydrothermal vents. These molecules, the ancestors of DNA and RNA, began copying themselves, sometimes imperfectly, and those imperfections, mutations, became the fuel of evolution. Over eons, simple life forms gave rise to complex organisms, each carrying the accumulated record of life's experiments. By the time the first primates appeared some 60 million years ago, the genetic code had already been honed by countless generations of natural selection. When modern humans appeared roughly 300,000 years ago in Africa, our genome was already a mosaic of ancient innovations. We inherited the molecular machinery of our primate relatives, yet subtle genetic changes gave us an unprecedented capacity for language, abstract thought, and cooperation. These were not the result of sudden leaps, but of small, gradual mutations. One change in a gene that affected brain connectivity, another that refined vocal control, and another that altered social behavior. The genome is the silent architect of civilization, shaping our destiny long before we could name it.
But how did we discover the code itself? The road to the genome's revelation was one of persistence and genius. In 1869, Swiss biochemist Friedrich Messier first isolated a mysterious substance from white blood cells, which he called nuclein. It would take decades before scientists realized this substance was DNA, the molecule of heredity. In the early 20th century, experiments by Oswald Avery and his colleagues proved that DNA, not protein, carried genetic information. Then, in 1953, James Watson and Francis Crick, building on the work of Rosalind Franklin and Maurice Wilkins, revealed the double helix structure of DNA. That image, two intertwined strands linked by rungs of base pairs, became an icon of modern science, symbolizing the unity of life and the universality of evolution. The genome story is not just a scientific journey, but a philosophical one. For centuries, humanity sought to understand itself through myth, religion, and history. The genome gave us a new kind of origin story, one written not in sacred texts, but in the language of biology. It showed that every human being shares 99.9% of their DNA with every other person on Earth, that the differences between us are tiny compared to what unites us. This revelation dissolved the illusion of biological race, proving that diversity is not a barrier, but a product of adaptation. The color of our skin, the shape of our eyes, the curl of our hair, these are minor variations sculpted by geography and climate, not markers of fundamental separation. Still, within that 0.1% difference lies the story of humanity's incredible adaptability. Our ancestors faced ice ages, droughts, diseases, and migrations that tested their resilience. Each challenge left subtle imprints on the genome: adaptations for digesting milk, resisting malaria, enduring high altitudes, or metabolizing different foods. These genetic signatures act as footprints on the trail of evolution. By reading them, scientists can reconstruct the routes our ancestors took as they spread from Africa to every corner of the planet. The genome is thus both a map and a memory. As we began to decode this map, new technologies transformed our understanding. Sequencing machines, once massive and expensive, became faster, cheaper, and more accurate. What took a decade and billions of dollars during the Human Genome Project can now be done in hours for less than the cost of a smartphone. This revolution opened the floodgates to genomic research, allowing scientists to compare human DNA with that of ancient fossils. From a few fragments of bone, entire genomes could be reconstructed, resurrecting long-lost relatives like the Neanderthals and Denisovans. These comparisons revealed that modern humans did not simply replace other hominins, we interbred with them, absorbing their genes and carrying them into the present. Today, people of non-African descent carry 1 to 2% Neanderthal DNA, while some populations in Asia and Oceania have Denisovan ancestry. These ancient contributions influence everything from our immune systems to our skin color, showing that our genome is a living fusion of ancient lineages. The revelation that we carry the DNA of extinct humans shattered the old notion of linear evolution. Instead, humanity's ancestry resembles a braided river, a network of branches that split and merge over time. The genome captures this fluidity, recording not just descent, but exchange, migration, and adaptation. It is a biological testament to our interconnectedness with all life, an evolutionary dialogue written in code.
The study of the genome also challenges our understanding of identity. If every human shares nearly identical DNA, what makes each person unique? The answer lies in the intricate patterns of gene expression, when and how genes are turned on or off. This is influenced not only by our inherited code, but also by environment, lifestyle, and even experiences. This field, known as epigenetics, has revealed that our genome is dynamic, capable of responding to the world around us. Trauma, diet, stress, and love can all leave marks on our DNA, changing how genes behave without altering their sequence. These epigenetic changes can sometimes be passed on to future generations, blurring the line between biology and experience. It means that the lives of our ancestors, their struggles and triumphs, may still echo in our genes today. To unlock the genome, therefore, is to awaken an ancient voice within us. It tells us that we are the sum of countless ancestors, each one leaving a trace of themselves in our DNA. It tells us that humanity's story is not one of separation, but of unity and shared heritage. It tells us that the blueprint of life is both fragile and enduring, capable of infinite variation, yet rooted in the same molecular alphabet that binds all living things. As science continues to decode this vast biological manuscript, we stand at the threshold of a new era, one in which we can not only read our genetic history, but also shape our genetic future. The implications are profound, touching on medicine, ethics, and the very meaning of being human. But before we can decide where we are going, we must fully understand where we came from. The genome is our compass, and in its sequence lies the story of evolution, migration, and memory. And so the journey into our ancient ancestry begins, not with bones or artifacts, but with molecules and code. It is a journey inward to the heart of what makes us human, and backward across millions of years of evolution. The genome is the ultimate storyteller, and its tale is still being written in every cell of our bodies.
The story of humanity's past is written not just in bones or artifacts, but deep within our DNA. Each of us carries a biological archive that holds evidence of journeys taken tens of thousands of years ago, when small bands of early humans left Africa to explore the unknown. Before written language or recorded history, before agriculture or cities, our ancestors carried the seeds of civilization inside their genes. The genome, in this sense, is not only a map of biology, but also a record of adventure, migration, and resilience. To trace these movements through genetics is to reconstruct the greatest journey ever taken, the journey of humankind across the world. The discovery that all humans can trace their lineage back to a common population in Africa was one of the most profound revelations in modern science. The out-of-Africa theory, supported by fossil and genetic evidence, tells us that around 60,000 to 70,000 years ago, a small group of Homo sapiens began migrating out of Africa, spreading gradually into Asia, Europe, and eventually to Australia and the Americas. These travelers carried only a fraction of the genetic diversity found in their African homeland, meaning that the further from Africa one goes, the less genetic variation one finds. This pattern, known as the serial founder effect, acts as a genetic echo of humanity's expansion. It is through this phenomenon that scientists can reconstruct the routes our ancestors followed and the timelines of their dispersal. Mitochondrial DNA, passed down exclusively through the maternal line, has been particularly useful in tracing these ancient movements. Because it mutates at a predictable rate, it acts as a molecular clock, allowing scientists to estimate when populations diverged. By analyzing mitochondrial genomes from people around the world, researchers identified what they call mitochondrial Eve, not a single woman, but a symbolic common ancestor of all living humans who lived in Africa roughly 150,000 to 200,000 years ago. Similarly, studies of the Y chromosome, inherited through the paternal line, point to a Y chromosomal atom, a male ancestor from around the same period. Together, these findings reveal that all humans share deep roots in Africa, even if our branches have spread across the globe. Yet, as humans journeyed out of Africa, they did not move in a straight line or in a single wave. Genetic evidence shows multiple dispersals, pauses, and returns. Some groups moved along the coastlines of the Arabian Peninsula into South Asia, adapting to new environments as they went. Others ventured into the Middle East and Europe, encountering Ice Age climates and interbreeding with local hominin populations such as Neanderthals. Meanwhile, separate waves of migration carried people into East Asia and eventually across the Bering Land Bridge into the Americas. Each of these movements left subtle traces in the genome, fragments of ancient DNA that reveal where our ancestors walked, what they endured, and who they met. One of the most revolutionary insights of modern genomics has been the discovery that humans are not purely descendants of Homo sapiens alone.
For many years, scientists believed that Neanderthals, who lived in Europe and Western Asia, went extinct around 40,000 years ago without leaving descendants. But when the Neanderthal genome was sequenced in 2010, it told a different story. Comparisons showed that modern humans of non-African descent carry between 1 and 2% Neanderthal DNA, evidence of interbreeding that occurred soon after humans left Africa. These genetic exchanges were not mere accidents. They may have provided adaptive advantages. Neanderthal genes influence the human immune system, skin pigmentation, metabolism, and even behavioral traits. In essence, the Neanderthals live on within us, their legacy intertwined with ours.
Further east, another mysterious hominin species left its mark, the Denisovans. Discovered from a few bone fragments in Siberia's Denisova cave, the Denisovan genome revealed an unexpected connection to modern humans. Populations in Melanesia, Papua New Guinea, and parts of Southeast Asia carry up to 5% Denisovan DNA. This suggests that our ancestors interbred with Denisovans somewhere in Asia before moving further east. Some Denisovan genes help modern Tibetans survive high altitudes, enabling efficient oxygen use in thin air. This adaptation is a remarkable example of how ancient DNA can directly shape modern survival. Our genome, therefore, is not just a record of the past, it is a living inheritance of ancient alliances. Beyond Neanderthals and Denisovans, scientists have uncovered traces of other, as yet unknown, human species in our DNA, what geneticists call ghost lineages. These are genetic signals that do not match any known fossil record, but suggest interbreeding with unidentified hominins. The existence of such lineages means that human evolution was not a simple tree, but a complex web of interactions. Each population, each encounter, added its verse to the genomic symphony that defines us today. The genome also chronicles our adaptation to changing environments. As humans settled in diverse climates, their genes evolved to meet new challenges. Populations in northern regions developed lighter skin to better absorb sunlight and produce vitamin D, while those near the equator retained darker pigmentation for protection against ultraviolet radiation. In regions where agriculture emerged, genes for lactose tolerance became widespread, allowing adults to digest milk. In malaria-endemic areas, genetic variations like sickle cell traits provided resistance to the disease, even though they carried health risks of their own. These examples show evolution in action, how natural selection shaped our genome to ensure survival in a constantly changing world. One of the most fascinating chapters in this genetic journey is the spread of language, culture, and innovation, phenomena that also left subtle genetic echoes. Genes that influence brain development, such as FOXP2, are thought to play roles in language ability. Variants in this gene distinguish humans from other primates, hinting at the genetic underpinnings of speech and communication. As our ancestors spread and formed new societies, cultural evolution began to intertwine with genetic evolution. The genome reflects not only biological change, but the rise of behaviors that define humanity: cooperation, art, music, and symbolic thought. Genetic research has also revealed how population bottlenecks, periods when human numbers drastically declined, shaped our genome. Around 70,000 years ago, the eruption of the Toba supervolcano in Indonesia may have caused a genetic bottleneck, reducing humanity to a few thousand individuals. The genetic evidence shows a sharp reduction in diversity from that period, followed by expansion. These bottlenecks left fingerprints in our DNA, patterns of reduced variation that scientists can trace across generations. They remind us of how fragile human existence once was, and how remarkable our recovery has been.
Another profound discovery has been the realization that genetic variation is not evenly distributed. African populations, being the oldest, hold the greatest genetic diversity on Earth. This diversity is a treasure trove for science, providing a clearer picture of early human evolution and disease resistance. Yet despite its significance, African genomic data remains underrepresented in research. As global genomics becomes more inclusive, the true depth of our ancestry continues to unfold.
revealing connections that challenge long-held assumptions about identity and origin. The genome also preserves evidence of ancient epidemics and immune responses. Many human genes evolved under pressure from infectious diseases. Traces of ancient viral infections are literally embedded in our DNA, remnants of viruses that inserted their genetic material into our ancestors' genomes. These endogenous retroviruses make up about 8% of the human genome, silent witnesses to countless battles between humans and microbes. Some of these viral fragments have been repurposed by evolution, playing roles in immune regulation and even placental development. Our genome, therefore, is both a battlefield and a record of past wars, a testament to survival through biological conflict. As we learn more, the genome's narrative becomes not only historical, but deeply personal. Through genetic testing, individuals can now trace their ancestral roots with remarkable precision. Millions of people have taken DNA ancestry tests, uncovering unexpected links across continents. These tests can reveal Native American ancestry in someone from Europe or African heritage in someone from Asia, reflecting centuries of migration, trade, and interconnection. The genetic evidence proves what history has long suggested. Humanity has always been mixed, always moving, always changing. The borders we draw are recent inventions. The genome knows no boundaries. But while personal genomics brings discovery, it also raises ethical and social questions. What does it mean to define oneself through DNA? Can genes tell the full story of who we are, or do they risk oversimplifying identity? The genome is a powerful tool, but it must be interpreted with care. Culture, environment, and experience shape human life as much as genetics do. To understand ancestry is to embrace complexity, not to reduce it. The more we sequence, the clearer it becomes that human evolution is ongoing. Our genes continue to adapt, to diet, to disease, to technology. Mutations that once conferred advantage may now be neutral or even harmful in modern contexts. For instance, genes that help store fat during times of scarcity now contribute to obesity in societies of abundance. The genome is dynamic, responding to our changing lifestyles and environments, writing new chapters even now.
Modern genomic science allows us to reconstruct ancient migrations in unprecedented detail. By comparing DNA from ancient skeletons to living populations, scientists can visualize the waves of movement that shaped the world. They can see the genetic imprint of the first farmers who spread agriculture from the Fertile Crescent into Europe, or the steppe nomads who brought Indo-European languages across continents. They can track the Polynesian expansion across the Pacific, or the Bantu migrations through Africa. Every strand of DNA is a line in a vast historical map, connecting the dots of human dispersal and exchange. Even our closest evolutionary cousins, chimpanzees and bonobos, help illuminate our story. Comparing their genomes with ours reveals the subtle differences that make us human. Just 1.2% of our DNA differs from that of a chimpanzee, yet within that small fraction lie the keys to language, technology, and art. The genetic divergence that began around 6 to 7 million years ago set in motion a chain of changes that led to self-awareness and civilization. The genome bridges these millions of years, linking us to the earliest primates and reminding us that humanity is not separate from nature, but a part of its grand continuum. Today, as scientists continue to analyze ancient DNA from ice cores, cave sediments, and fossils, new surprises emerge almost daily. Genomic archaeology has turned prehistory into a living science. Ancient genomes recovered from Neolithic farmers, Bronze Age warriors, and Paleolithic hunters are rewriting the history of migration and interaction. They reveal that populations have risen, mingled, and vanished countless times, and that every human alive today carries echoes of those forgotten peoples. The genome is a mosaic of memory, a biological archive of all who came before. In the end, tracing our genetic footprints shows us something profound, that humanity's story is one of unity through diversity.
Beneath our different languages, cultures, and appearances lies a shared code that binds us all. The genome does not lie. It shows that every person is a distant cousin, every lineage a branch of the same tree. Our migrations, intermixing, and adaptations have made us resilient and creative. The genome reminds us that our strength lies in connection, not division. That the human story is a single narrative written in countless variations of the same code. As the map of our ancestry becomes clearer, so too does our sense of belonging. To know our genome is to see the invisible threads that link us to ancient Africa, to the Neanderthal caves of Europe, to the Denisovan mountains of Asia, and to the first explorers who crossed oceans and ice. It is to understand that we are, in every cell, children of the same earth, travelers on a timeless journey, carrying the legacy of millions of years within us.
The human genome is more than a sequence of letters. It is a living library of information, a symphony of biological instructions that governs every cell, every thought, and every heartbeat. Hidden within its spiraling structure is the story of how we grow, heal, think, and evolve. To understand it is to translate the language of life itself. But this language is not straightforward. It is full of codes within codes, regulatory switches, and hidden messages that even today, decades after the first genome was sequenced, we are still struggling to fully comprehend. The journey to decode the secrets within the human genome is one of the most fascinating scientific adventures of our age, one that merges biology, technology, and philosophy in a quest to understand life's blueprint. At its core, the genome is composed of DNA, a molecule built from 4 chemical bases: adenine, A, thymine, T, cytosine, C, and guanine, G. Arranged in specific sequences along chromosomes, these bases form genes, which act as instructions for making proteins. Proteins are the machinery of life, enzymes, hormones, and structural components that perform the countless functions required for survival. Yet, surprisingly, only about 1.5% of our genome actually codes for proteins. The remaining 98.5%, once dismissed as junk DNA, is now known to play critical roles in regulating gene expression, structuring chromosomes, and protecting genetic integrity. The so-called junk is in fact a vast landscape of switches, enhancers, and non-coding RNA molecules that fine-tune the orchestra of life. The complexity of this regulatory system is staggering. A single gene can produce multiple proteins through a process called alternative splicing, where different segments of RNA are pieced together in various ways. This means that while humans have roughly 20,000 to 25,000 protein-coding genes, fewer than some plants and worms, our capacity for complexity arises from how those genes are used, not merely how many we have. It is like having a limited alphabet, but an infinite capacity for poetry. The genome's elegance lies in its efficiency, in the way it uses a simple language to produce unimaginable diversity. But decoding the genome is not just about identifying genes, it's about understanding how they interact. Genes rarely act alone. They operate in networks, responding to environmental signals, hormones, and even other genes. A change in one region of the genome can ripple through these networks, influencing traits or diseases in ways that are difficult to predict. The Human Genome Project provided the map, but the human genome is a living system, dynamic, adaptable, and responsive. To interpret it requires not just sequencing, but comprehension, a task that has given rise to the field of systems biology. Perhaps the most profound discovery of genomic research is how similar all life is at the molecular level. Humans share about 98.8% of their DNA with chimpanzees, 85% with mice, 60% with fruit flies, and even 50% with bananas.
This genetic overlap reveals a deep evolutionary kinship that transcends species. The same genes that control cell division in yeast play roles in human cancer. The genes that shape fin development in fish helped form our limbs. Evolution is not a process of creating from scratch, but of modifying and reusing successful designs. In our genome, one can see the fingerprints of millions of years of experimentation by nature, a record of life's continuous creativity.
The genome also holds within it the mechanisms of inheritance and change. Every generation introduces slight variations, mutations, that can alter a gene's function or expression. Most mutations are neutral, some are harmful, and a few confer advantages that natural selection favors. Over time, these small changes accumulate, shaping species and populations. The human genome, therefore, is both stable and flexible, a structure that preserves essential information while allowing for innovation. This dual nature explains how humans could remain genetically similar to our ancestors while developing new abilities like abstract thought, language, and technological imagination. One of the most intriguing revelations of the genomic age is the role of epigenetics, changes in gene activity that do not alter the DNA sequence itself. Chemical tags, such as methyl groups, can attach to DNA or to histone proteins around which DNA is wrapped, turning genes on or off. These tags act like annotations on the genetic script, directing how and when the underlying code should be read. Environmental factors, diet, stress, toxins, even emotional experiences can influence epigenetic patterns, meaning that our lifestyles can leave molecular imprints on our genes. Astonishingly, some of these marks can be passed on to offspring, creating a bridge between biology and experience. Epigenetics shows that we are not just prisoners of our DNA, we are participants in its expression. Epigenetic inheritance offers a biological echo of memory. Studies in animals show that trauma, famine, or environmental stress can alter gene expression in ways that persist across generations. In humans, evidence suggests that descendants of people who endured extreme hardships, such as the Dutch hunger winter during World War II, show distinct epigenetic signatures, affecting metabolism and health. These findings blur the line between genetic and environmental inheritance, revealing that the genome is not fixed but alive, constantly writing and rewriting itself in dialogue with the world. At the same time, decoding the genome has opened vast new frontiers in medicine. Understanding genetic variation allows doctors to predict disease risks, tailor treatments, and even prevent illness before it begins. This is the promise of personalized or precision medicine, healthcare based on an individual's genetic makeup. By analyzing a person's genome, scientists can identify mutations linked to cancer, heart disease, diabetes, or neurological disorders, enabling early interventions. Drugs can be chosen or designed based on how specific genes affect metabolism, reducing side effects and improving efficacy. The genome transforms medicine from reactive to proactive, from one-size-fits-all to uniquely tailored care. One striking example of this revolution is in oncology, where sequencing tumor genomes reveals the mutations driving cancer growth. Instead of classifying cancers solely by their location in the body, doctors now classify them by genetic signatures. Two patients with lung cancer may require completely different treatments because their tumors have distinct mutations. Genomics allows for therapies that target the molecular roots of disease rather than just its symptoms. It represents a shift from treating illness to understanding it, to reading the body's internal story in molecular detail.
Yet, as we gain power over the genome, we face profound ethical dilemmas. The same tools that allow us to prevent disease also enable us to edit genes, to alter the very instructions of life. Technologies like CRISPR-Cas9 have made gene editing precise, efficient, and accessible. With CRISPR, scientists can cut DNA at specific sites and insert or remove sequences, effectively rewriting the code of life. This has revolutionized biology, offering potential cures for genetic disorders like cystic fibrosis, sickle cell anemia, and muscular dystrophy. But it also raises questions about where the line should be drawn. Should we use these tools only to treat disease, or should we also enhance human abilities, intelligence, strength, appearance? The genome, once a mirror of our past, now becomes a canvas for our future. These questions are not merely scientific, but philosophical. Editing the genome means assuming responsibility for evolution itself. For billions of years, life evolved through random mutation and natural selection. Now, humans hold the pen. This power challenges our notions of identity, ethics, and destiny. The genome, which once told the story of where we came from, may soon tell the story of what we choose to become. Are we ready to write that next chapter? To answer that, we must first deepen our understanding of how the genome works in harmony with its environment. Our genes are not destiny. They are potential. Whether a gene is expressed depends on countless factors: diet, behavior, social interaction, and culture. Even identical twins who share the same DNA can differ in personality, health, and lifespan because of these environmental influences. The genome provides the script, but life directs the performance. The Human Genome Project and its successors have shown that the genome is not an isolated entity, but part of a complex ecosystem. Within each human cell live trillions of microorganisms, the microbiome, whose genes outnumber our own by 100 to 1. The microbiome interacts intimately with our genome, influencing digestion, immunity, mood, and even cognition. In many ways, we are not individuals, but symbiotic collectives, walking ecosystems whose health depends on balance between host and microbes. The study of this interplay, sometimes called the second genome, is redefining what it means to be human. Another revelation from genomic research is that evolution never truly stops. Even in the modern world, our genes continue to change. For example, populations in East Africa and Northern Europe independently developed lactase persistence, the ability to digest milk into adulthood, within the last 10,000 years. This is an extraordinarily recent adaptation, showing how quickly culture and genetics can influence each other. The rise of agriculture, urbanization, and technology continues to exert new pressures that shape our biology. Our genome is still evolving, even in the age of artificial intelligence and digital life.
But evolution is not always progress. Some mutations that once conferred survival advantages now predispose us to modern diseases. Genes that helped our ancestors store fat during times of famine now contribute to obesity and diabetes in societies of abundance. Variants that strengthened immune responses against infections may now increase the risk of autoimmune diseases. The genome is a historical record, not a perfect design. It carries the compromises of evolution, the remnants of ancient battles between survival and adaptation. The more we decode the genome, the more we realize that its story is not one of perfection, but of resilience. Every error, every mutation is a potential for innovation. Life thrives on imperfection, because imperfection allows diversity, and diversity allows survival. The genome teaches us that change is not a flaw, but a force, the very essence of evolution. Understanding this complexity has required immense computational power. Genomics has become a data science as much as a biological one. The sequencing of a single genome produces terabytes of information, requiring advanced algorithms, artificial intelligence, and machine learning to interpret. The marriage of biology and technology has given birth to bioinformatics, a discipline that translates raw genetic data into meaning. Computers now help us identify patterns invisible to the human eye. Networks of genes, evolutionary relationships, and potential targets for medicine. The genome has transformed science into a dialogue between code and consciousness. Even as we explore the intricacies of the human genome, we find that its beauty lies in its universality. The same genetic code, the same four-letter alphabet, underlies all life on Earth.
A bacterium, a tree, a whale, and a human all read the same molecular language. This unity suggests that life, in all its forms, shares a single origin. Every living thing is a variation on the same theme, a different verse in the same biological poem. To study the genome is to glimpse the shared ancestry of all existence. Yet, for all our advances, vast portions of the human genome remain mysterious. Thousands of genes have functions we do not yet understand. Non-coding regions harbor elements that may regulate development, intelligence, or longevity, but their mechanisms are elusive. Some parts of the genome appear to be dark matter, sequences conserved across species, yet defying explanation. The more we learn, the more we realize how much remains unknown. The genome is both an answer and a question, a mirror reflecting our progress and our limits.
The act of decoding life has also reshaped our relationship with nature. Once, humans saw themselves as separate, as masters of the natural world. The genome reveals that we are deeply entangled with it. The same molecular machinery that builds a fern also builds a human being. Evolution connects us not only to animals, but to all living things. Our genome carries traces of ancient viruses, bacterial symbioses, and evolutionary experiments. It is a biological tapestry woven from countless threads of life. This realization carries moral weight. To manipulate the genome is to intervene in a system older and wiser than ourselves. It demands humility. For every gene we understand, there are thousands whose functions remain unknown. The genome teaches patience, that life is not a machine to be engineered, but an ecosystem to be understood. Every discovery brings both power and responsibility, and the future of genomics depends on how wisely we wield that power. The language of life is subtle, poetic, and profound. Within its syntax lies not only biology, but philosophy. The genome asks us to consider what it means to be human, to be both product and producer of evolution, both observer and participant. It shows that identity is not static, but a process, unfolding through time, shaped by both ancestry and choice. In the double helix, we see ourselves reflected, fragile, creative, and endlessly curious. And so, decoding the human genome is not merely a scientific pursuit. It is a spiritual one. It reconnects us with our origins, reminding us that we are part of an unbroken continuum of life stretching back to the dawn of Earth. The same chemical bonds that once sparked in ancient oceans now dance within ourselves. To read the genome is to listen to the voice of evolution, the whisper of life itself, still speaking through us.
Every living human carries within them a vast biological chronicle, a story millions of years in the making. The human genome is not only the record of our individual lives, but also the archive of our species' entire evolutionary journey. It holds the echoes of our transformation from primitive primates into sentient beings capable of art, science, and love. Every gene is a whisper from our ancestors, every mutation a note in the grand symphony of evolution. To study the human genome is to stand at the intersection of biology and destiny, gazing into the ancient code that made us who we are. When we examine the genome through the lens of evolution, patterns begin to emerge, patterns that trace the slow sculpting of human nature. Some genes carry the marks of ancient battles against disease, others the fingerprints of adaptation to diet or environment, and still others the mysterious signatures of cognitive evolution. The genome tells us that the story of humanity was never random. It was guided by pressure, survival, and creativity. Evolution did not design perfection, it designed possibility. Within our DNA lies both the fragility of life and the potential for greatness.
At the root of our evolutionary success lies the brain, a structure that consumes nearly 20% of our body's energy and sets us apart from all other species. The genome reveals how this extraordinary organ came to be. Small mutations over millions of years gradually enhanced neural development, synaptic connectivity, and cognitive flexibility. One of the most critical of these was the duplication of the sRGAP2 gene, roughly 2.5 million years ago, a change that allowed for the growth of longer, more complex neural dendrites, boosting brain plasticity. This single genetic event may have paved the way for the rise of tool use, communication, and imagination, the hallmarks of human intelligence. The genome preserves such turning points in its sequence like fossils in digital form, revealing how biology laid the foundation for consciousness. Other genes contributed to our cognitive evolution in subtler ways. Variations in the FOPX2 gene, often dubbed the language gene, distinguish humans from other primates and are associated with speech and linguistic ability. Mutations in this gene in modern humans can lead to speech disorders, underscoring its importance in vocal learning. The appearance of this variant in our evolutionary timeline roughly coincides with the development of symbolic thought and cultural complexity. Through the genome, we see how a few molecular tweaks transformed grunts and gestures into the words, poems, and philosophies that define civilization. Yet, as the genome shaped our intellect, it carried forward remnants of our animal past. Ancient reflexes and instincts still influence behavior, encoded deep in our DNA. Hormonal systems regulating fear, mating, and aggression evolved to ensure survival in a world of predators and scarcity. Modern life often activates these same pathways in environments where they no longer serve their original purpose. The genome thus reveals both our progress and our constraints, that the same genes that made us adaptable hunters now drive stress in the digital age. Evolution is not a clean slate, but an accumulation, a constant negotiation between the past and the present.
The genome also reveals the story of our bodies, how they adapted to a changing planet. As Homo sapiens spread across continents, natural selection fine-tuned genetic variations for survival. Populations in colder climates developed genetic variants that improved fat storage and heat conservation, while those in tropical regions evolved efficient sweating and resistance to heat stress. In the Andes and Himalayas, genes affecting red blood cell production allowed survival in thin, oxygen-poor air. Even the way we process food bears the imprint of evolution. The amylase gene, which helps digest starch, became more prevalent as agriculture spread, reflecting a shift from hunting to farming. The genome remembers every meal humanity has ever eaten, every climate it has ever endured. But not all genetic changes came from adaptation. Some were the result of chance. Neutral mutations, those neither beneficial nor harmful, accumulated over generations, serving as molecular clocks that mark the passage of evolutionary time. These mutations are the quiet background music of evolution, the steady rhythm that measures the flow of generations. By analyzing these neutral changes, scientists can estimate when populations diverged, reconstructing the human family tree with astonishing precision. The genome is both the diary of evolution and its timekeeper. Among the most profound revelations of modern genomics is the understanding that our species is not alone in its ancestry. Long after our split from other hominins, humans continued to exchange genes with their ancient cousins. The Neanderthal and Denisovan contributions to modern genomes, once seen as anomalies, are now recognized as essential threads in our biological fabric. The Neanderthal genes enhance immune responses and keratin production, influencing hair and skin. Others affect mood regulation and sleep cycles, reflecting adaptations to life in northern latitudes. The Denisovan EPSA1 gene, which aids oxygen use, remains vital for Tibetan highlanders. These inherited fragments remind us that humanity's story is one of mingling, not isolation. Our genome is a collective memory, a shared mosaic of vanished species that still live on through us.
Yet, the genome is not only a record of evolution's successes, but also of its costs. Many diseases that afflict humanity today are, in a sense, side effects of evolution. The same genetic flexibility that allowed us to adapt can also go awry. Disorders like sickle cell anemia, cystic fibrosis, and Tay-Sachs disease arise from mutations that persist because they once provided advantages in certain environments. The sickle cell trait, for example, confers resistance to malaria, ensuring its survival in tropical regions despite the risk of disease. Similarly, genes that once boosted fertility or immune function can, in modern contexts, predispose people to cancer or autoimmune conditions. The genome, in its complexity, carries both the light and the shadow of our evolutionary past.
The study of ancient DNA has also transformed our understanding of human evolution. From fragments of bone and teeth, scientists can now extract and sequence entire genomes of individuals who lived tens of thousands of years ago. These genetic time capsules reveal the faces of our ancestors in unprecedented detail. We can trace how farming spread through Europe, how hunter-gatherer groups mixed with incoming migrants, how empires rose and fell within our DNA. Each ancient genome is a letter from the past, adding nuance to the grand story of humanity's evolution. The technology that makes this possible, next-generation sequencing, has turned prehistory from speculation into data. We can now reconstruct not just where people lived, but how they were connected by blood and kinship across millennia. Through these discoveries, the genome also reveals the intimate connection between biology and culture. As humans began to shape their environment through technology, their environment began to shape them in return. The domestication of plants and animals, the invention of cooking, and the rise of cities all exerted genetic pressures. Cooking, for instance, made food easier to digest, reducing the need for large jaws and digestive tracts, changes reflected in our anatomy and genes. The shift to agriculture altered diets, favoring genetic variants for metabolizing grains and milk. Urban living increased exposure to pathogens, selecting for robust immune systems. Culture and genetics evolved together, each driving the other forward in an intricate dance that continues to this day. Perhaps nowhere is this co-evolution more evident than in the relationship between humans and disease. Epidemics have repeatedly shaped the genetic landscape of humanity. The Black Death, smallpox, tuberculosis, and influenza all left marks on the genome, favoring genes that confer resistance. The CCR5 Delta 32 mutation, which offers protection against HIV, likely spread centuries ago due to its defense against earlier viral outbreaks. Our immune system, one of the most complex products of evolution, is a living monument to these ancient struggles. Every antibody, every immune receptor is a product of evolutionary improvisation, sculpted by countless generations of conflict with microbes. The genome records this invisible war, encoding the memory of every pathogen that ever tested human survival. And yet, even as the genome bears the scars of the past, it also illuminates the path to the future. The study of genetics allows us to foresee how humanity might continue to evolve. Modern medicine has reduced the power of natural selection, but new selective pressures have emerged: urbanization, pollution, changing diets, and technology itself. Our reliance on digital tools, artificial environments, and global interconnectedness may shape the future genome in ways we can barely imagine. The boundaries between biology and technology are blurring as humans begin to augment their own evolution through science.
This interplay between nature and innovation raises profound questions about what it means to evolve. Are we still products of natural selection? Or are we now guiding our own genetic destiny? The genome shows that evolution never stops. It merely changes form. As our ancestors once adapted to savannas and glaciers, so we now adapt to skyscrapers and screens. The pressures are different, but the process endures. Evolution is not a relic of the past, it is the constant hum beneath the surface of life. The genome also serves as a mirror for the diversity of humanity. A diversity born of adaptation, migration, and chance. Each human population carries a unique constellation of genetic variants, shaped by history and geography.
yet all are part of the same continuum. Genetic studies show that differences between populations account for only a tiny fraction of total human variation. The concept of race, so entrenched in social consciousness, has no biological basis. Our genome exposes it as an illusion. The real story is one of shared ancestry and overlapping lineages, of unity expressed through variation. The genome unites us all in a single continuous human family. Understanding this has immense moral significance. In a world often divided by culture and color, the genome speaks a language of unity. It tells us that every person on Earth descends from the same small group of ancestors in Africa. It shows that we are all kin, branches of a single tree, leaves of the same ancient root. To read the genome is to see beyond the boundaries that separate nations and tribes, to recognize the profound interconnectedness of life. The code that builds a poet in Paris is the same code that built a shepherd in Ethiopia or a monk in Tibet. The differences are surface ripples upon an ocean of shared humanity. The genome also forces us to confront the paradox of knowledge, that the more we learn, the more mysterious life becomes. We can read the sequence of our DNA, but we still cannot fully explain consciousness, creativity, or the will to survive. The genome provides the hardware of existence, but the software, the essence of thought and emotion, remains elusive. Somewhere between molecules and mind lies the spark of humanity, a mystery science continues to chase. Yet even without complete understanding, the genome offers profound insight. It reminds us that our greatest gift is curiosity, the drive to explore not only the stars, but the code within. In the last 2 decades, scientists have begun to explore the possibility of resurrecting lost genes, of reactivating ancestral traits dormant within our DNA. Some researchers study the reintroduction of extinct species genes into modern relatives, a field known as de-extinction. Others seek to revive ancient immune defenses or enhance disease resistance by reawakening evolutionary pathways. These experiments blur the line between science and philosophy, between memory and creation. They raise difficult questions. Should we restore what evolution erased? Should we edit our own genomes to prepare for future challenges? The answers will shape not only our biology, but our moral identity.
As we peer deeper into our genetic past, one thing becomes clear. The genome is not just about survival, it is about connection. Every gene is a thread woven into the fabric of life. The same code that sustains a human sustains the oak tree, the whale, and the eagle. Evolution, seen through the genome, is not competition but collaboration, a great network of shared ancestries stretching across time. Life is not a hierarchy, but a web, and we are one strand among billions. To understand the genome is to understand our place within that web, to see ourselves as both products and stewards of an ancient process. The echoes of evolution resonate in every heartbeat, every breath, every thought. Our genome sings the song of life, one that began long before humanity and will continue long after us. It is a song of resilience, adaptation, and unity, of molecules becoming minds, of chaos giving birth to consciousness. To unlock the genome is to hear that song in full, to recognize that in our DNA lies not only the story of what we were, but the promise of what we might yet become.
The story of the human genome does not end with discovery. It begins there. For the first time in history, our species has turned its gaze inward, deciphering not just the universe around us, but the universe within. The 21st century marks a profound transition in the human journey, the moment when we ceased to be mere subjects of evolution and became its conscious stewards. With each strand of DNA sequenced and every gene mapped, we gain not only knowledge, but power. The power to heal, to alter, and perhaps even to redefine what it means to be human. Yet with that power comes responsibility. For within the double helix lies not only our ancestry, but our destiny. When scientists first completed the Human Genome Project in 2003, it was hailed as a triumph equal to the moon landing. For the first time, humanity held in its hands a complete map of its own genetic architecture. 3 billion base pairs written in the language of life. But mapping was only the beginning. Decoding its meaning would take decades more. And even now, we have only begun to understand the vast complexity hidden within that code. The genome is not a static blueprint, but a living, dynamic system. It changes with time, environment, and experience. Epigenetic switches turn genes on and off in response to diet, stress, or trauma, revealing that our biology is as fluid as our lives. The genome is a dialogue between inheritance and environment, a bridge between what we are given and what we become. This revelation transformed our view of evolution itself. No longer a process confined to deep time, evolution now unfolds within the span of individual lives. Our choices, what we eat, where we live, how we think, leave chemical marks on our DNA, sometimes passed to our descendants. Trauma experienced by one generation can ripple through the next, altering gene expression in subtle but lasting ways. This field, known as epigenetics, bridges the ancient with the modern, suggesting that the echoes of famine, war, or love can resonate across centuries. The genome is not merely inherited, but it remembers. In understanding that, humanity has stepped into a new moral and scientific frontier, the power to edit its own genome. Techniques such as CRISPR-Cas9, discovered in the past decade, have revolutionized biology. With CRISPR, scientists can precisely cut and replace genetic sequences, correcting mutations that cause disease or introducing beneficial traits. What once seemed like science fiction is now routine in laboratories, editing embryos to prevent hereditary illnesses, engineering immune cells to destroy cancer, or designing crops that resist drought and pests. Each breakthrough promises salvation and raises profound ethical questions. If we can rewrite our genetic fate, should we? Who decides what constitutes a better genome? Where does healing end and enhancement begin?
The temptation to design perfection is as old as myth. Yet the genome reminds us that perfection is an illusion. Evolution thrives on diversity, on variation, on the unpredictable interplay of chance and necessity. To erase imperfection is to risk erasing possibility. Many traits once seen as flaws, resilience under stress, creativity born from struggle, or genetic variations conferring disease risk, also contribute to the richness of the human experience. Our genome's strength lies in its imperfection. It is the rough edges that make life adaptable, the random mutations that fuel progress. The future of genetics must therefore be guided not by control, but by compassion, a recognition that our biological diversity is our greatest inheritance. Still, the potential for good is immense. Genetic medicine is already transforming healthcare. Diseases once deemed incurable are being confronted at their molecular roots. Disorders like cystic fibrosis, muscular dystrophy, and certain forms of blindness are now treated through gene therapy, replacing faulty DNA with healthy sequences. Personalized medicine, tailoring treatments based on an individual's genetic profile, promises to revolutionize how we fight cancer, diabetes, and heart disease. The genome is becoming both a diagnostic map and a medical arsenal. With every discovery, we move closer to a world where illness is not an inevitable part of human existence, but a solvable riddle written in code. Beyond medicine, genetics is reshaping how we understand identity itself. In the past decade, millions of people have explored their ancestry through direct-to-consumer DNA testing. These services, once novelties, have unveiled hidden connections across the globe. Lost relatives reunited, migrations traced, forgotten heritages restored. For many, the results challenge long-held assumptions about race, culture, and belonging. The genome dissolves boundaries drawn by history, reminding us that bloodlines are fluid,
not fixed. We are all composites of countless ancestors, woven together through time. The genome tells a story of unity that transcends borders, a living proof that humanity is one extended family. But the power to know our genetic story also brings unease. What happens when the code that defines us becomes data to be stored, sold, or manipulated? As genetic information enters the digital world, questions of privacy and ethics grow urgent. DNA, once hidden in the sanctity of ourselves, now sits on servers owned by corporations and research institutions. It can reveal not only our identity, but our health risks, propensities, and even psychological traits. The genome, once sacred, has become a commodity. This new reality forces humanity to confront the meaning of ownership. Do we own our DNA, or does it own us? The challenge of the genomic age is not just scientific, but moral. How to balance discovery with dignity, progress with privacy. At the same time, genetic research is expanding our understanding of what it means to be alive. By comparing the human genome with those of other species, scientists have identified the deep genetic roots we share with all life on Earth. Over 98% of our DNA is identical to that of chimpanzees, but we also share genetic sequences with birds, fish, and even bacteria. These connections reveal that life is not a hierarchy, but a continuum, a shared lineage stretching back to the dawn of the planet. The same molecular language that writes the human body also writes the tree and the whale, the butterfly and the bacterium. The genome is the great unifier of biology, proof that all living things are variations on a single theme: survival, adaptation, and transformation.
As the field of genomics advances, scientists are beginning to look beyond Earth. Space agencies now study how the human genome responds to the challenges of space travel, radiation, isolation, and microgravity. Prolonged time in space can alter gene expression, changing how the body repairs DNA, produces blood, and regulates immunity. Understanding these changes is essential as humanity prepares for long-term missions to Mars and beyond. The genome, once shaped by the forests and savannas of Earth, will soon face the evolutionary pressures of the cosmos. In that sense, our species is standing on the threshold of a new evolutionary chapter, one in which we carry not just the memory of Earth, but the blueprint of life itself into the stars. Yet, amid this cosmic ambition, the genome continues to reveal the intimate, fragile beauty of human existence. It teaches us that life is a collaboration, not a competition. Within our bodies, genes cooperate in intricate networks, balancing creation and destruction, growth and decay. The same principle governs ecosystems, societies, and civilizations. The genome whispers an ancient truth. Survival is not achieved through domination, but through balance. When we disturb that balance, through greed, neglect, or violence, we endanger not only our species, but the entire web of life that sustains us. To unlock the genome is not only to read the code of life, but to assume guardianship over it. The ethical dimensions of this guardianship are immense. As gene editing, cloning, and synthetic biology advance, we approach questions once reserved for philosophy or religion. Can we create new forms of life responsibly? Should we resurrect extinct species or alter existing ones? What rights, if any, should artificial or hybrid organisms possess? Each discovery stretches the moral fabric of civilization. The genome forces us to confront the nature of creation itself, to ask whether we are caretakers of life or architects of its future. The line between evolution and engineering blurs, and the choices we make now will echo for generations to come. Amid these challenges, the genome also offers hope, a path toward healing not only bodies but societies.
The realization that all humans share 99.9% of their DNA is a reminder of our shared humanity. The genetic differences that divide us are statistically insignificant, yet culturally magnified. The genome reveals that beneath our skin tones and languages, we are virtually identical, variations on a single theme composed billions of years ago. This knowledge carries moral weight. It dismantles the biological basis of racism and prejudice, replacing division with understanding. The genome, in its quiet objectivity, calls for empathy. It reminds us that the fight for equality is not just political, but biological, written into the very code of who we are.
The journey of unlocking the genome also mirrors the human condition itself, a quest for meaning in the face of complexity. Every discovery opens new mysteries. Every answer deepens the wonder. We learn that the genome is not deterministic, it is possibility embodied. A gene may predispose, but it does not predestine. Choice, environment, and chance shape the final outcome. Within this interplay lies the essence of freedom, the power to transcend our biology through knowledge and will. The genome tells us that we are not prisoners of our past, but participants in an ongoing evolution. We are the first species to hold its own genetic script and the first to choose how the story continues. As we look ahead, humanity faces a pivotal question. Will we use this knowledge to heal or to harm, to unify or to divide? The genome itself offers guidance. It teaches that diversity strengthens resilience, that adaptation requires cooperation, and that every mutation, like every idea, carries the potential for renewal. The next phase of evolution may not be written by nature alone, but by our collective choices. The genome gives us the tools. Conscience must give us direction. In laboratories around the world, researchers now dream of engineering the human genome for the next age of exploration, to resist disease, endure harsh environments, or extend lifespan. Some envision a future where aging is optional, where memory can be preserved through molecular repair, where humanity becomes a self-evolving species. Others caution against hubris, reminding us that evolution's wisdom cannot be easily replicated. For billions of years, natural selection refined life through balance and trial. To take control of that process is to inherit both its power and its peril. The genome's greatest lesson may be humility, the understanding that life's complexity exceeds our comprehension, and that our role is not to dominate it, but to harmonize with it. In the end, the human genome is more than a scientific map, it is a philosophical mirror. It reflects our past and hints at our potential. It speaks of ancestors who crossed deserts and oceans, of tribes who survived ice and fire, of minds that dreamed of gods and galaxies. Every cell carries that heritage, the echo of 1,000,000 generations whispering through time. To unlock the genome is to listen to those whispers, to realize that we are not separate from nature, but its most self-aware expression. The atoms in our blood were forged in stars. The code in our cells was written by life itself. The genome is the universe made self-reflective. As the story of the genome unfolds, humanity stands at a crossroads of wonder and responsibility. We are the first species capable of rewriting its own evolution, yet we remain bound by the same moral questions that have guided philosophers for millennia. The double helix, elegant and eternal, reminds us that knowledge without wisdom can destroy, but wisdom guided by compassion can transform. The future of the genome is not about control, it is about understanding, coexistence, and the celebration of life's endless creativity. And so, as we look into the spiraling code of our existence, we find not just science, but poetry, the poetry of survival, adaptation, and unity. The genome does not speak in words, yet it tells the oldest story ever told, the story of becoming.
From the first spark of life in the primordial sea to the rise of civilizations and the dreams of spacefaring humans, that story continues, encoded within us. The genome is the bridge between what was and what will be, between memory and possibility, between humanity and eternity.
In the end, to unlock the human genome is to unlock ourselves, our fears, our hopes, our boundless curiosity. It is to see that we are not the end of evolution, but its latest expression, still unfolding, still learning. Our DNA carries the wisdom of the past and the promise of the future. It whispers a single truth across the ages, that life, in all its forms, seeks not perfection, but understanding, not immortality, but connection. And perhaps that is the true legacy of the human genome, the realization that within every one of us lies a fragment of the universe, striving to know itself.
The journey never ends. The story ends here, but the journey of knowledge never stops. Subscribe to our channel, stay inspired, and be ready, because the next chapter begins soon.